Hello everyone. This is a bit of a different format than what we usually do, but it's something that I wanted to test around with, play around with, see if something like this would be interesting to do. Trying to change the format up a little. I was thinking, I realized that the best way to get me talking about a lot of scientific topics is either to have someone with me, but obviously that's not going to happen all the time, or to basically just have it right in front of me to talk about, and then I can go off on any whatever tangent that I want to. So that's kind of what today's going to be like. We're gonna go through like a few scientific news articles, or at least news articles about scientific topics, because I want to, kind of share what's out in the world and also learn if I don't know really what the article's talking about at all. It'll be a good way of sort of teaching and learning at the same time. But yeah, so let's get started. So this first one I got, it's on science.org, and it's titled "Ancient Virus May Be Protecting the Human Placenta." And I'll play around with the format of this. So far I'm thinking, just pull an article up. I haven't even read it, no preparation, nothing like that. So I think that'll just be more fun getting into it. Of course, that'll probably mean that we'll hit some duds. I'll just be like, oh, this was actually extremely boring. Next article. But that's just the roulette wheel we play. But let's see. This one says, pathogen that became embedded in our genome millions of years ago may ward off other viral invaders. Okay. And I'll kind of, if there's anything I don't understand or can't really explain, then that's when we look it up. We sort of read something else, and then we're like, oh, okay, so that's how that works. And it'll just make for an easier way to understand. So let's see, about 30 million years ago, a virus infected our primate ancestors, and one of its genes got trapped in their genomes. So in case you're not aware, the way viruses work, actually, let's see if we can pull up a picture. Mechanism, sure. So there's a few different ways that viruses can replicate, essentially. God, if I remember the terminology. What was it called? Translation replication. Let's see. Is it transmission mechanism? Sounds wrong. Virus, replication. This all help me keep up my vocabulary. Let's see. Replication. Is it intrinsic? Let's see, viral replication, blah, 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 blah. No, there's a specific term I'm looking for. And if I can just find that. Let's see, viruses can either replicate in a bit of a temporary sort of way, or in a more permanent sort of way. And there is a specific term for that. Let me just throw things out there. Intrinsic, that doesn't sound right. Two types of replication, lysogenic cycle, nope. Not what I'm looking for. Well, regardless. We'll just see what we can find. Anyways, virus replication can either go into the cell, and depending on which level it decides to replicate at, whether it decides to essentially throw itself into our genome, or if it just uses some other mechanism that's sort of in the cell. then it will, if it puts itself into our genome, then we just make the virus for it. Like, it doesn't have to do anything else because it just hijacks our DNA replication, the way that our cells replicate. So let's see. This might be kind of good. So yeah, anyways, releases the contents, then has the mRNA. No, this looks like the other way. Anyways, so that's how that works. Yeah, so basically what this is saying is that a long time ago there was a virus, and it went ahead and slapped some stuff into our genome. Humans kept replicating, and then humans kept, I don't feel like there's a better word for that. I mean, it's not wrong, but, humans kept mating, and over time, that just became a staple of the genome, it seems. And now we just have that. So let's see, over time this viral gene became domesticated and territorial. It helped primates fight off other viruses by preventing them from entering cells. The invader, known as suppressin, Supian, is still around today. It's still helping us out. A new study reveals that this viral turncoat might help the placenta protect embryos from viral infection. Okay. It's basically saying that whatever this virus is, let's see, what is this? Retroviruses such as HERV-K, shown here in red, can permanently integrate their genetic material into human genomes. But basically, sounds like suppressin, basically. I'm curious what it means by territorial. But helps primates fight off other viruses by preventing them from entering cells. Okay. We'll dig into it, and then we'll see if there's more to go off of from there. It's a beautiful story supported by very strong experiments, says Yula Kisa, an evolutionary biologist at the University of Colorado Boulder, who is not involved with the work. Okay. Alright. Then who was involved with the work? Where was this at? Finding more such viral genes, she says, might help us harness or boost our inherent antiviral properties without the need to develop new drugs or vaccines. There are all of these elements we already have in our genome. I don't... I can also throw in all the opinion pieces I want in here. But I feel like... I don't know about without the need to develop new drugs or vaccines. Maybe a drug to, like, boost these antiviral properties. Possibly, but, I think our bodies are just gonna do what they do without any kind of drug or vaccine. Let's see. Viruses that embed their genetic material in our genome are known as retroviruses. HIV. Yeah, it's a retrovirus, but there's a specific term, and I forget. HIV is probably the most famous example. Once integrated into our genes, it hijacks our body's cellular machinery to produce more viruses. They infect sperm cells, or oocytes, the precursors of eggs. Their genes become part of our DNA and can be passed on to our offspring. So this is true. This is why HIV is kind of a bitch. Is because the fact that it can be passed down through one's lineage, that can create a whole shitload of problems. Yeah, let's see. Just because it's gonna kill me, let me run with retrovirus real quick. Let's see. Scripted. I just want to find that one term. Here, let's see. It's a virus inserts a DNA copy of its RNA genome to the DNA of a host cell that invades. Thus changing the genome of that cell. Yep, uses. We have a process called transcription, which leads from DNA to RNA. But viruses will use reverse transcription, which essentially takes their RNA, then puts it into our DNA. Let's see, integrase, no. Damn. Guess I just won't find it. That's alright. Oh, that was the wrong thing. So let's see. Pass on to our offspring. Once a snippet of viral DNA becomes embedded in our genome, it's known as an endogenous retrovirus. Is that it? Was it endogenous? I mean, there's endogenous and exogenous, but I don't think that's the process itself. But endogenous just basically meaning that it's within the cell, and exogenous meaning it comes from essentially out of the cell or uses something, doesn't use the DNA in the cell. About 8% of the human genome consists of ERV sequences that have been trapped in our DNA ever since they infected a human ancestor billions of years ago. These genes have lost their original viral function over time, but that doesn't mean they're useless. So also, we have evolved with obviously tons of bacteria over time, and that's actually why a lot of bacteria is 100% necessary for our survival. Like, we need certain bacteria to live. It's also kind of like the theory of, let's see, I forget what it is, but the theory of mitochondria actually being a bacteria. Let's see. Endosymbiotic hypothesis for origin of mitochondria. Basically, mitochondria at one point kind of looks like it was a unicellular organism. And that's why, small picture. So most of the organelles in the cell will have like one membrane around it. But the theory is because mitochondria actually have a double membrane around it, almost like it was taken into the cell. So the theory is, one day, at one time it might have been like a unicellular thing, and maybe it had some other purpose, possibly. Over time, it sort of got this symbiotic relationship with human cells or animal cells or whatever, what have you. And so we just have mitochondria now that live in our cells. So sort of the same way with a bunch of other bacteria that we've gotten over time. And I guess like some endogenous retrovirus DNA, it might not have its original function, but it can still serve a function for us. So let's see, to find out which ERVs may still be active in the human body, molecular biologist Cedric Shoddy? I will pronounce so many names wrong, by the way. Of Cornell University and his colleagues scanned the human genome for ERV sequences they suspected might encode proteins. Found 1,570 sequences, about half of which seemed to be doing something in human tissues. One ERV turned up in the human genome known as suppressin, which encodes a protein produced in the placenta in early human embryos. Okay, hold on. Let's see what this does. Okay. Yeah, let's see what this gene does first of all. Okay, so it's an endogenous retrovirus group, 48 member 1. See, many different endogenous retrovirus families are expressed in oral placental tissue at high levels, suggesting that endogenous retroviruses are functionally important in reproduction. Kind of implying that because there's so many retroviruses that are in placental tissue, then they must have a purpose to be there. There must be some sort of function for them all. This gene is part of an endogenous retrovirus provirus that has placenta-specific expression. I always forget, what is provirus again? Genetic material of a virus has incorporated into and able to replicate with the genome of a host cell. Okay, so basically just the DNA of the virus, I guess. The protein coded has the characteristics of a retroviral envelope protein, but is truncated and lacks a transmembrane domain. Protein inhibits cell fusion by competing with Syncytin 1 for binding to a cell receptor. Okay. So basically, kind of like this article is getting at, it just sounds like this is a retrovirus sort of a gene. Okay. Yep, spines receptor, known as ASCT2, the same receptor in another ERV-derived protein called syncytin, uses to form links between cells. In its former life as a retrovirus, syncytin could fuse with cell membranes to enter cells. Its modern form allows placenta to form during fetal development by fusing cells together. Okay, so basically with cells dividing, I guess this helps keep things together? Or syncytin at least did that? The evolution of the placenta might not have been possible without it. You could argue that without retroviruses, you wouldn't have mammals, says Wilkin Johnson, a virologist at Boston College. It's almost like life wouldn't have evolved as it did. Which, I don't really know too much about the evolution of placenta and where that came in from. But you know, I believe it. Like I said, viruses and bacteria shaped a lot of the way that animal and down the line human evolution came into play. But let's see if we can find that out. How can I search this up without searching anything bad? Placenta evolution origin. I'm curious when it first came in. Placentas evolved from pre-existing tissues such as the uterus and allantoic membrane. Hence, to understand the evolution of placental functions, it's important to identify the genes expressed in the ancestral tissue from which the placental structure is derived. Let's see. So this is an article on Nature Ecology and Evolution. The placenta is a model for understanding the origin and evolution of vertebrate organs. We'll probably just go through abstract. I don't think we'll go through the full thing. Hold on a second. Check something real quick. Okay. How organs originate and evolve is a question fundamental to understanding the evolution of complex multicellular life forms. Vertebrates have a relatively standard body plan with more or less the same conserved set of organs. So basically, vertebrates usually have the same amount of organs. Placenta is a comparatively more recently evolved organ, derived in many lineages independently. It's basically different animals have kind of evolved the placenta independently recently. I say recently in a maybe the last few million years sort of sense. Using placentas as a model, we discussed the genetic basis for organ origins. Okay, so this is looking into kind of how organs evolve and how they come about. Which if I were to guess, I would probably just say random mutation, because that's a lot of evolution, is just random mutation. And then, you know, it might make something where it's like, oh, this is beneficial to the body. Like, organisms can survive with this. We showed that the evolution of placentas occurs by acquiring new functional attributes to existing tissues, changes in the patterning and development of tissues, and the evolution of novel cell types. So yeah, again with the sort of mutations. So getting new functional attributes to existing tissues. So tissue's already there, but it kind of gains a new function in a way. We argue that a diversity of genomic changes facilitated these physiological transformations, and that these changes are likely to have occurred during the evolution of organs more broadly. So, I guess trying to be like, most organs came about this way. Finally, we argue that a key aspect to understanding the evolution of origin of organs is that they are likely to result from novel interactions between distinct cell populations. So it also depends on the cell type for these things to evolve. Okay, so this is more going into not so much the placenta, but as, I guess kind of as it says, using the placenta as a model for understanding where organs can come from. Let's see, what animal first had a placenta? Sometimes it can be kind of hard to find the answers to the questions you need, because they'll be muddled with other random things that Google's like, oh, you want this, like the normal layperson wants. And I'm like, no, I don't. No, I don't, Google. Don't question me when I search for things. Let's see, which were the first animals? True placental mammals, the crown group, including all. I guess we can just use the Wikipedia article. Placental mammals are one of 3 extant subdivisions of the class Mammalia. So I guess as opposed, well, I'm trying to think, do kangaroos? Let's see. Name is something of a misnomer, considering that marsupials also nourish their fetuses via placenta, though for a relatively briefer period giving birth to less developed young, which are then nurtured for a period inside the mother's pouch. Okay, so even marsupials have placenta. Interesting. Placental mammals are anatomically distinguished from other mammals by... Let's see, as of 2020, the genome has been sequenced for at least one species in each extant placental order in 83% of families. Okay. Okay, evolutionary history. Placental mammals, the crown group, including all modern placentals, arose from stem group members of the clade Eutheria, which had existed since at least the Middle Jurassic period, about 170 million years ago. These early Eutherians were small, nocturnal insect eaters with adaptations for life in trees. So it's these guys, Eutheria. They consisting of all Therian mammals, they're more closely related to placentals than to marsupials. Okay. Oh, that's that. Let's see. Anyways, back to it. ASCT2 is also an Achilles heel for mammals. Viruses called type D retroviruses use it to bypass cellular defenses to sneak into cells and cause a variety of diseases in many animals, including non-human primates. None, though, are known to affect humans. So this is another thing where viruses and bacteria can actually also, I mean, kind of like COVID, they use your own structures against you. So I can use COVID just because it's a, relatively recent example. But the COVID virus uses a protein called ACE2 that is on the surface of, I guess I don't know if it's all cells or if it's just cells around blood vessels, but anyways, it's on the cell surface, and this protein is usually used for like blood pressure and stuff. But COVID, it's kind of like a lock and key mechanism where the protein on the cell surface is the lock, and then COVID kind of acts as the key. Even though it's not the key that was meant for there, imagine someone create another key or a copy of a key, or just sort of made like a crude version of the key. And so the cell goes, you fit my protein ACE2, like come on in. And then COVID goes, don't mind if I do. So that's how a lot of different viruses and bacteria will get into your body, because they'll just kind of use that mechanism to be like, let me come on in. So I guess it sounds like this same thing for this, where it's used to bypass other defenses. Let's see. Shaw says this could have presented a major challenge to early animals if they didn't have a way to protect themselves against these retroviruses. Safeguarding the placenta would be particularly important, as retroviruses that infect an embryo early enough in development could get into sperm and egg cells. True. Definitely had to be a lot of defenses for the human body as far as reproduction. Otherwise, yeah, obviously, embryos would just get fucked up by different diseases. When Fasha and his colleagues experimentally infected human placental cells with retroviruses, they found that supin competed against the pathogens by blocking ASCT2 receptors, making it impossible for viruses to enter the cells. So this is one method of defense that cells have against things such as COVID. And well, I guess I can go down that rabbit hole. So again, imagine the lock and key mechanism, but imagine there's like a stopper in the lock so that you can't put another key in. So there's no way the door can open from the key because something's already taking the place of the lock. So it sounds like this, the suppressin basically blocked this receptor, and that prevented any viruses from getting in. Here, we'll go through this paragraph at least. The cells seem to turn suppressin on when they detect a virus, suggesting that it encodes an antiviral protein, the researchers report today in Science. So, you know, with that line of thinking, you may ask yourself, oh, well, like, why didn't we do that for COVID, or like do that for any other virus thing? Like, why don't we just block the virus, block the receptor that it needs? Well, that's because a lot of times receptors have an actual use. There's something else that needs to actually be put into those receptors. And if it's blocked from one thing, then it basically blocks everything. Although there are some cases where I believe we have had like sort of half blocked proteins, but then that can open up a whole nother can of worms because then different, again, lock and key mechanism metaphor, different key can go in there and then cause other mayhem. But it, unfortunately isn't just like easy one kind of solution to stopping viruses and whatnot. Johnson notes it's unclear whether suppressant actually blocks any viruses in people. Although type D retroviruses can infect macaques and other primates, none of them appear to infect humans. The authors have got the means and the motive. The problem is the opportunity, he says. It's all guilt by association. I think what he's getting at, and this does happen in science a lot, is kind of a classic case of correlation versus causation. We might be like, with an increase in suppressin in these cells, it seems like the viruses don't infect them, so therefore suppressin is stopping the infection. But unfortunately that might not be the case, where it's like, oh, actually suppressin does this or that, and then down the line something else blocks it, maybe it's just like a random happenstance or through some other mechanism. So it's also why I dislike a lot of journal articles kind of leaving out those details on things. Where they'll be like, this causes this or whatever, and it's like, well, no, if you read the article or sort of didn't try to just summarize it, like A doesn't lead to B. Actually, it's like A leads to C, D, E, F, G, which then leads to B. Yeah. Yeah, just one of the very annoying things of trying to learn what causes what in a body. Let's see. So Johnson notes it's unclear whether suppressant actually blocks any viruses in people, although okay, yeah. Still, Johnson praises the paper. He notes that it's possible that suppressant evolved to block a virus that is now extinct, or perhaps it is so good at its job that it prevents enemy viruses from ever gaining a foothold. You know, because it's one of those things of kind of like, well, how do you really test to see if the human placenta is being blocked? Or is blocking these viruses in particular, whatever the paper used. Skessy adds that it's technically impressive the authors managed to figure out which... Did we talk to the authors at all? It sounds like... This article makes it sound like we just... kind of talk to people that know about the article. Hold on, is this the paper? Let's see. Yeah, I guess I would have to go looking for the paper then. Let's see. We're almost done with this one. Let's see. Impressive, the authors managed to figure out which receptor these retroviruses were using to enter cells. She's currently working on a similar study, mapping domesticated ERVs in the human genome, and has found about 30 that appear to be important to the human immune system. It's amazing how our own body makes all of those little antivirals, she says. It's very impressive how much our body does do without us even knowing. Frichelle's team now plans to work through the dozens of other active retroviruses that they identified. Points out that although suppressant is recognized as a functional human gene, 99% of the other active ERVs they found just look like unimportant junk DNA, but looks can be deceiving. This is true. We say there's a lot of quote-unquote junk DNA, and a lot of times we find out like, oh, this actually had a purpose, like this spot or whatever. So you know. That's another thing, like scientists can't really just write off a chunk of DNA as junk DNA. If they're like, oh, we just need to get rid of this junk DNA to do, you know, to accomplish this outcome or whatever. It's like, there has to be a lot of testing to go through before we can actually be like, oh yeah, no, definitely, this is useless. Then you say, there's this treasure trove of potential proteins, he says. There's at least the potential that these treasures have interesting activities for medicine or physiology or development. Okay, pretty good. Yeah. Ancient virus may be protecting the human placenta. Okay. So I get now, basically to summarize, virus, I mean, honestly, I don't even want to say in humans, but yeah, virus infected primate ancestor, stayed in the DNA, and now today it helps block viruses from getting into the placenta, if that is the case. Pretty good. Pretty good. All right. So, I got a few articles, and maybe we can do more. But we'll see. I mean, the first one that alone took like half an hour explaining everything and doing things. So I think that's pretty good. Let's go with the next one. So I saw this morning, and I was like, this sounds so fucking silly to me. But scientists invent first vaginal on a chip. The first vaginal on a chip replicates the cellular environment in the vagina. And I was just like, you know, I think I read like a second just to be like, what the fuck? But it makes sense, but the fact that they call it vaginal on a chip, But if I understand correctly, let's see, microfluidic organ chip platform developed at the Weiss Institute and subsequently licensed to a company called Emulate Inc. But basically, I think it's like just cells on this little plate somewhere, like human cells, and then also the bacteria that is found in the vagina. And then that's what, you know, just to sort of imitate the environment, I guess. But the fact that they just straight up label it as vagina on a chip is so fucking silly. But let's see what this is actually about. Scientists have developed the world's first vagina on a chip, a small device that contains live human cells. Let's zoom in on a little bit. Okay, so yeah, live human cells and replicates the cellular environment found inside the vaginal canal. By adding bacteria to the device, actually named the vagina chip. Okay, so I guess they have these organ chips, and then whatever organ is, they just say X chip. So this is the vagina chip that we're talking about. Researchers can study how different microbes affect the health of the vagina. The team reported in a new study published November 26 in the journal Microbiome. Is this the actual paper? Vaginal microbiome hosts interactions modeled on a human vagina on a chip. Okay, yeah. Sweet, and we can look through the whole thing if we want. So let's see. They can also test how different drugs and probiotics change the composition of the vaginal microbiome, the community of microorganisms living within the canal. So ridiculousness of this aside, this is actually great technology just in general for any organ or whatever, because you know, being able to test things on people is such a slippery slope. You know, we can't always just be like, hey, take this drug, see if it affects them or whatever. You know, we have to very carefully do this, or like very conservatively choose our like demographics and whoever we want to test it. But with something like a little chip to imitate the organ, or at least the environment in the organ, we can kind of maybe have an idea of how that all will work. So it's pretty good idea. Let's see. Vaginal microbiome plays an important role in regulating vaginal health and disease and has a major impact on prenatal health. First author Gautam Mahan, a former researcher at Harvard University's Weiss Institute for Biologically Inspired Engineering, said in a statement, "Our human vagina chip offers an attractive solution to study host microbiome interactions and accelerate the development of potential probiotic treatments, which work by introducing beneficial bacteria into the vagina, said Mahan, who now works at the organ on a chip company Emulate Inc. in Boston. Okay, so I guess Emulate Inc. now does all the organ on a chip stuff. But yeah, that is interesting, being because you can do the same. You know, in theory, it would probably be a bit more, but you know, a term you probably heard a lot in the past few years, like microbiome, the bacteria that you have in your gut and everything. So kind of the same thing. Shows like the microbiome of the vagina in this case. Here, I think this is their. So this is kind of a thing where some journals and stuff are trying to push scientists to be doing video abstracts or video like presentations with their paper, and to include that, since a lot of things are digital nowadays. I'm kind of like, feels annoying to have to create a whole video for it or whatever, but let's turn that off. Let's watch the video abstract, see what they have to say. Vaginal microbiome plays an important role in vaginal health and disease. For example, certain lactobacillus species dominate in a healthy vagina, while disruption in the microbial status quo is associated with disease. Therapies to manipulate the vaginal microbiome are therefore promising for preserving vaginal health. Also, I don't know, it feels like a what was it, like a clickbait kind of video or something, where it's just like stock footage and they just throw shit over it. Let's see, but no preclinical models have been able to faithfully replicate the vaginal epithelial microenvironment for experimentation. Yeah, it can be complicated too. Until now. So I'm curious, yeah, I'm sure they'll go into it, but I'm curious about this design of like why have the line across? Why not just, why not just a plate of all the bacteria and cells? Let's see, researchers at the Wyss Institute at Harvard just developed the vagina chip, a 3D in vitro model featuring a functional vaginal epithelium. Okay, so let's see. When assemblages of beneficial vaginal microbes like Lactobacillus carpatus are grown on chips, the epithelial cells remain healthy. Okay. Vaginal epithelium, porous membrane, chromofibroblasts. Okay, so I guess the fibroblasts are on one side, and then they put the vaginal epithelial cells on top. That's just basically, vagina cells, porous membrane, so that things don't technically get mixed together, but they will still be able to communicate if, to be able to send signals to each other. Okay. Let's see, in addition, beneficial D and L lactate accumulate, normal vaginal pH is maintained, and pro-inflammatory molecules are downregulated. Okay, so this is saying that even though they include the bacteria and things, there is still a lot of manual things that have to be done to take care of this sort of thing. So that's, which, makes sense. That makes sense for basically any cell work in a lab. Another quick thing I want to say real quick, just a personal thing. This is fine and all, but if there is any time, not necessarily this, but if someone tries to convince you something with like some sort of engineering problem or something, or some sort of infrastructure problem, and they give it to you in a 3D like movie style format of like, here's our solution, here's how it will work, and then show it moving in a 3D space or 3D CG, like that's how you know that shit's a scam. That's how you know to absolutely not trust whatever bullshit is being peddled through. Again, not this in particular, because they showed the chip. We can literally see it up there. Yeah, I mean, it's right there. Hello, killer. Yes. I'm right in the middle of talking about the vagina chip, where this is something that was developed to where scientists can actually imitate the environment of the inner vagina and still be able to test different probiotics and whatever on it. But I'm doing good. How are you? I'm glad you're able to make it. Yeah, we're just in the middle of watching the video abstract that they have for this. Yeah, but again, it sounds like they have other chips they have. Other organ chips. But I guess, easy to get clicks on an article that literally has vagina chip in the title. But let's see, okay, so still have to maintain other things manually though. Can't just take care of itself. In contrast, some groups of harmful bacteria like Gardnerella vaginalis are grown on the chips, the epithelial cells become damaged. Okay, so basically, makes sense. I think it said, yeah, beneficial cells are grown on there. Obviously, the or beneficial bacteria grown there, obviously the cells are fine. Harmful bacteria grown, then it dies. And the pH and pro-inflammatory cytokine levels increase. Okay, so they can see how the pH is affected through the vagina chip. Consistent with pathological changes in human vagina with an unhealthy microbiome. So that is interesting. I guess they had to take some actual people, or at least data from before, to sort of compare it to be like, how close is this to an actual human vagina? Like, how comparable is this actually for us to be able to use it? Although additional confirmation is needed, the findings demonstrate that the new vagina chip is a promising preclinical model. Should be pretty nice, honestly. You know, there's been a lot of super shitty experimentation on women's vaginas and stuff just throughout history. So it would be very good if, you know, people weren't being taken advantage of in a study where they had no idea they were applying for it or whatever. Also this, or I kind of hate when articles are just like, yeah, this is sort of the preliminary thing, like additional confirmation is needed, but like, looks good. So it's just like, who knows, maybe this whole thing will fall through and it won't be so great. Let's see, that can be used to clarify vaginal epithelium, microbiome interactions, and test new therapies. Okay. Yeah, and we got the paper right here if we want to look into some more details. So you reached a square and partner with Springer Nature, and this is on a microbiome? Okay, so maybe this video was put together. Actually, no, they had the video right here. Yeah, it says video abstract. So there's our video abstract. Okay. So the scientists allowed these cells to multiply for 5 days in the device before adding the sex hormone estradiol, a form of estrogen. A hormone change which genes are switched on in the tissues and triggered the production of mucus, as would occur in vivo. Interesting. Okay, so it even produces its own mucus. Okay, so they just had to add estradiol. With their device complete, the team ran several tests with bacteria commonly found in the vagina, namely several strains of lactobacillus bacteria. Studies suggest that these microbes make up more than 70% of a healthy vagina's microbiome, according to the License 2 statement. Researchers found that the lactobacillus bacteria successfully produce lactic acid in the vagina chip, thus driving down the pH of the tissue inside. A healthy vagina typically has a pH of 4.5 or less, meaning it's acidic, it'll burn your dick off, and this acidity helps burn, or, I'm joking, but helps prevent the growth of harmful bacteria that could cause disease. Yeah, a lot of places in our bodies, the pH is very specific, or at least around specific levels for defenses. So, you know, a lot of times whenever there's some sort of like diet or something that's like, oh, your stomach or like your whatever shouldn't be this pH, it should be this pH. A lot of times that's complete horseshit. Like, you want your stuff to be as close to the natural pH as possible. You don't want to try to change any of that stuff. In addition, adding lactobacillus bacteria reduced the number of inflammatory molecules circulating in the tissues. Okay. God, you know, I didn't really work on inflammation, but I hate it so much because there is simultaneously both good and bad inflammation. So it can be such a pain in the ass sometimes, because sometimes in some studies it's like, like, you know, if things are going good, it produces good inflammation. If things are going bad, it produces bad inflammation, and it can be sort of confusing which is which, you know? But I guess in this case, yeah, just talking about any kind of like bad inflammation. Having run experiments with good bacteria, the team did the same with bad bacteria, meaning bugs associated with bacterial vaginosis, common vaginal infection caused by the overgrowth of specific bacteria. These bugs include Gardnerella vaginalis, Prevotella bivia, and a Topobium vaginae, which, and when introduced into the vagina chip, all three bacteria cause the pH of the device to rise, along with the number of inflammatory molecules and damaged vaginal cells. Okay, so that basically they could, yeah, what they're implying here is they could have both a healthy vagina chip, but also a vagina chip with BV, and then try to introduce drugs to the slightly unhealthy vagina chip to create a better, treatment or whatever. So that's pretty good. BV increases the risk of STDs such as chlamydia and gonorrhea, both of which can negatively affect future fertility by triggering a harmful inflation, according to the CDC. If developed during pregnancy, BV also raises the risk that a baby will be born prematurely or at a low birth weight. I didn't actually know that. The new vagina on chip could help scientists develop new and better treatments for BV. Sexual health physician Akuta Nori of St. George's University of London, who is not involved with the study, told Scientific American. Again, we went through one article already. It talked about like an ancient retrovirus essentially protecting placenta during growth. And they kind of did the same thing where they're like, yeah, we talked to this one scientist who was not involved with this study at all and got their opinion. And I'm just like, what? Like, why? Why'd you ask this person in particular? But it's whatever, I guess. It's fine. This is an opportunity to bring women's health into the modern times using modern technology, Nori said. This chip offers an advantage over testing therapies in lab animals like mice, whose vaginal microbiomes greatly differ from humans. Also very hard to do with animals sometimes. Sometimes animals don't have the same, like, gut biome or microbiome or whatever. So animal studies might not be good enough for these types of things. In addition, it's historically been difficult for drug developers to recruit human patients for BV treatment trials, partly due to patients' concerns over safety, experts told The Times. Yeah, it's because they're honestly... I could probably go into it or try to find something, but there are so many treatment trials for drugs or whatever that, of course it's a little bit better, but like, say the 1930s, for example, like, we'll just tell when, a lot of people still couldn't read that well, and we would just tell them like, oh, this is, we have this trial to do this or like to fix this or whatever, and they'll be like, okay, and then it's a trial to do something completely different or just has like no actual scientific founding, and then as a result, like, people or like women can get fucked up, and it's just like, it's like, well, what the fuck? And they'll be like, oh, sorry, you agreed to it. It's like they can't even read. You just lied to them to sign this thing. That said, Oregon on chip technology does have its limitations, so it cannot perfectly capture all the factors that influence the human vaginal microbiome, but the vagina chip is a good first step towards further research. Okay. Interesting. So there you have it, the vagina chip. Again, this says Oregon chip. I wonder, here, let's take a look at this. At their website here, Emulate Inc. I actually am curious what other kind of organs they have chips for. Human relevant models for complex biology. Okay, so we got brain chip, colon intestine chip, duodenum intestine chip, kidney chip, liver chip, lung chip. Okay, so they do have like multiple. They are doing multiple organs, which is... okay, no, I see. The brain chip is under development. Which actually, now that I think about it, yeah, that'd be fucking wild if they had that one set and ready. Lung chip also under development. Let's look at kidney chip. Let's see, predicting drug-induced kidney toxicity and drug-drug interactions. Actually, you know what? No, the liver chip. That sounds like it'd have some pretty good studies. Combines 4 human cell types and dynamic microenvironments to support in vivo like gene expression, functionality, and physiology. Validated applications include general and mechanistic toxicity evaluation, as well as assessment of AAV-based gene therapy, transduction, efficiency, and safety. I don't remember what AAV-based gene therapy is. Adeno-associated virus vector is a platform for gene therapy delivery. Oh, okay, so using viruses as a way for gene therapy. I see. Okay, same thing. Where it's got this little, yeah, it's got this membrane between them. On the bottom, you have the immune cells and the endothelial cells. And then on the top, you can have the hepatocytes, and at least in the liver chip's case. Interesting. And this is talking about what, how different cell studies can, like, essentially what the liver chip has over different cell studies and things. Technical. But yeah, neat. That's pretty cool. I hope it actually works out for future studies and stuff. The human emulation system. Actually, hold on. Now, how much would it man, I hate this. So a lot of science stuff does this, where if you want to try to figure out like, oh, how much does something like this cost? They won't tell you outright. Like, you have to contact them and then get a quote from them. And I'm just like, I'm like, why can't you just have like a general price, or at least a price range or something, and then get a quote? You know, very, very annoying. Let's see. Maybe they'll say here. Because yeah, I mean, you know, if the chip costs like, well, it depends. You know, if it's like each chip, like if each chip costs several $100s or maybe even like thousands of dollars, then for the moment, a lot of people probably won't use it because they'll just be like, your shit's expensive. Fuck that. But yeah, that's the vagina chip, I guess. It's the full paper. Let's see if there's anything interesting in here. Human vagina chip. So this is one skill you kind of develop doing a lot of papers. We can sort of, you know, I'm not gonna actually need to read it that much, but essentially learning how to quickly go through a paper and then sort of learning like, okay, what information in this do I actually want to talk about in like a meeting or something. Let's see. Talking about the good bacteria, lactate production. Oh yeah, I forgot about the music. Oops. We finished listening to the abstract and I totally forgot about the music. Ooh, actually, I wonder in the methods, do they say the cost? Let's see. Nothing. Okay. That's fine, doesn't say. Alright, yeah, that's the vagina ship. So I saw this one as well. I was like, oh, this just seems silly. So this is, are these bumblebees playing with toys? First example of insects seemingly having fun with objects adds to evidence for emotions. The dudes are just hanging out, playing with the balls. Okay. Playtime isn't just for children. Lab cat bumblebees, you know, because there's a lot of talk of like, can like insects or fish or whatever actually feel pain / emotions or whatever? And I feel like that's always a question that's kind of in the air. Because I think partly it's kind of yes, partly kind of no. So it's just kind of weird. Let's see. Lab cat bumblebees roll small wind balls around for no apparent purpose other than fun, a new study reveals. Finding supports evidence that bees experience pleasure, researchers say, highlighting the importance of protecting them in the wild and treating them well when they're kept in hives. It's super cool, says Elizabeth Tibbetts, an evolutionary biologist at the University of Michigan, Ann Arbor, who was not involved. Again, I feel like I'm gonna run into this a lot when reading through articles. Is it's just gonna be like, yep, we talked to this biologist. They weren't involved in the study at all. Let's see, we usually think about insects as being so different that they lack sophisticated behaviors, but not everyone is convinced the behaviors in fact play. Well, just thinking about it, it is kind of hard to, you know, if your theory is, oh, they're playing, well, it's kind of hard to, and this is like a lot of people who will come up with an idea for something, and they'll be like, see, and say, well, okay, there's no easy way to disprove that, you know, that they're playing. Like, unless we can get some sort of, oh, he's got a little number on him. I didn't realize that they were numbered. Okay. Yeah, it's hard to disprove a theory like that. Like, how can you say they are not playing? You would have to come up with some other reason. It can be kind of difficult. In animals, play helps the brain develop. Foxcubs pretend and fight to learn social skills, for example. And dolphins and whales jump and spin even without predators around. A study in 2006 described behavior in young wasps that looked like play fighting, but whether other insects play hasn't received much research attention. Lars Chitka, a behavioral ecologist at Queen Mary University of London, and author of the recent book The Mind of a Bee, and colleagues stumbled upon the new evidence by accident. The team was studying how bumblebees learn complex behaviors from their comrades by training the insects to move wind balls to specific locations. If a bee moved a ball to the right place, it got a sugary treat. The researchers noticed that some bees moved the balls even when no reward was offered. It just seemed to like going back to them, fiddling with them, rolling all over the place, Chico says. Samadi Galpage, then a master student at QMUL, was fascinated by the bees' many behaviors. I couldn't help falling in love with them, she says. I stayed on to do a PhD to learn more. Galpage set up what was essentially a single-story apartment. Okay, single-story apartment for the bees. At one end was the nest, which had a single entryway to a recreation center. Far end of the rec room connected to a cafeteria with an all-you-can-eat supply of pollen and sugar water. The recreation center was where the experiment took place. This room had two play areas on either side, each with wooden balls that were slightly bigger than a bee. The balls couldn't roll into the path, so the bees had to literally go out of their way to play with them. Okay, that's good, because for me, I wasn't sure. I was like, it sounds like the recreation center is next. Yeah, it's like next to the cafeteria to be able to get the sugar water, so they would have to go through there. In the first experiment, Galpage and colleagues immobilized the balls on one side. To get to the buffet, the bees had to walk through the recreation room and past the ball areas, and they clearly preferred the side with the balls that rolled, entering it 50% more times than on average. That suggests the bees like the movement, not just round objects. Well, to be fair, so is there something besides balls put in there? Because I don't... Does that really suggest that they like the movement and not just round objects? Oh, I see. There's a ball area that did not roll and a ball area that did roll. I understand. Measuring how many times each bee rolled a ball. See, this is good because this is sort of the kind of eliminating causation versus correlation that you have to do. Where it's like, okay, these bees are on these balls. Do they like the movement, or do they just like round things? Measuring how many times each bee rolled a ball. See video above. The team found that some only did so once or twice, but others rolled them up to 44 times in a day. A repetition implies they are enjoying the activity. I would also be curious about changing the balls to a different material / maybe other colors on them. Even though I know bees see in different colors than us. To confirm that rolling these balls is indeed the bee's knees. Very good. The Go page set up another experiment with a new nest of bumblebees. Like the previous design, bees leaving the nest pass through a chamber to get to the food. For the first 20 minutes, the chamber was colored yellow and contained the wooden balls. Then Galpage swapped in a blue chamber without balls. He alternated the colored chamber 6 times, trained the bees to associate the color yellow with the presence of the balls. Finally, Galpage gave the bees a choice between entering a yellow or blue passageway without any balls in sight. The bees favored yellow, about 1/3 more chose it, presumably because they associated it with the pleasurable sport of ball rolling. The researchers report this week in Animal Behavior. When the team reversed the colors and repeated the experiment, they got similar results. I think it provides really good evidence that rolling balls is rewarding, Tippett says. Okay, so also very good. Again, I was thinking like, okay, did they like, is it actually that they like the color yellow or do they like the balls? So it seems like they did test for that, which is good. The experiment also revealed that young bees were more likely to roll ball than were older bees. This reflects how play changes with age in birds and mammals. Young animals are more likely to play. Researchers think their developing brains might benefit from this experience, for example by strengthening neuronal connections involved in muscle coordination. The bee brain is also better able to form new connections early in life, before workers first leave the nest to begin foraging. So basically just give bees some balls to roll. Helps their minds. Galpage is now investigating whether ball rolling improves the ability of adult bees to efficiently harvest nectar from flowers. That would be interesting. Gordon Burkhardt, an expert in animal behavior at the University of Tennessee, Knoxville, says the careful design of the experiments has convinced him the bees are indeed engaging in play. I think this is a great paper. Well, cool. Sergio Pellis, who studies animal behavior at the University of Lethbridge, isn't fully sold. Even though the experimenters gave the bees a clear path to a reward, he wonders whether the balls still might be triggering the housekeeping behavior that the bees use to remove dead bees and other debris from their nests. To make a stronger case that bees can experience pleasure, he says, it would help to have more examples of play. I guess that's true, yeah. Maybe include some other sort of shape or something. Even if the bees are indeed playing, it's unclear whether they would do so in the wild. It's possible that bees in the lab have more opportunities for play because they are protected from predators and don't need to gather food, Chitka says. Competition in the wild is tough and might not provide bees with the luxury to set aside time to manipulate objects just for fun. Yeah, this is kind of true. Yeah, because if they have the time, if they have the time to just mess around, do whatever, maybe they would get bored. So it's, it's fair points. And again, why it's really hard to have some sort of lab experiment versus like wild animals or whatever, because we honestly just don't know. We wouldn't fully know the difference if we don't do it. Because play implies a capacity to experience emotions, the researchers say documenting in insects could have ethical implications. Insects are increasingly being reared for animal feed, and there are no regulations governing their welfare, Galpage notes. Honeybees are also known to become stressed and more vulnerable to disease and colony collapse when industrialized beekeepers transport them long distances on trucks to orchards and vast fields without diverse flowers nearby, Trika says. The researchers hope their findings might also motivate greater empathy for and protection of wild insects. Okay, so yeah, they kind of covered their bases here about everything. But for the time being, we can absolutely just be like, you know what? Those bees are playing and they enjoy it. That one was a little short. Let's see what else we got. Mitochondria depiction. Telescope school, nice. Sees the universe anew. Strange tentacled microbe may resemble ancestor of complex life. Research gets a boost in final 2023 spinning agreement. Sweet. Mars had long-lived magnetic field, extending chances for life. Progressive Union strike controversial deal with University of California. Male wasp fend off attackers with penis stingers. We don't have to. Oh, come on. Reach my limit. Hate that shit. All right, I guess we won't go on science anymore. Yeah, see if they really have any kind of play behaviors. It would be interesting, right, to see if they have any. I mean, I guess there's some way for us to check to see if there's dopamine or serotonin or something similar. Let me see if I can pull up something else. Man, I hate that kind of stuff where it's just like, oh, you only get, X amount of articles. So fuck you, pay us. If you want to read more. Ugh, just found an article I definitely don't want to go down, but it's Twitter changed science. Weapons, now it's in turmoil. Microblogging platform has transformed research communication, but its future is in doubt. Yeah, I don't know if Twitter had that much of a change on researching. Alright, sounds good. Let's see, we could build space cities and asteroids like in sci-fi with this wild concept. Okay. Let's take a look. Even though I'm a little skeptical, because like I was saying earlier, if there's some sort of technology that's conceptual and they give it to you in some fancy CG demonstration, then I'm gonna be honest, I don't have big faith in it. Like, I need to see something solidly built. Yeah, artist's conception of a futuristic city inside an asteroid near a planet. But let's see. A pandemic-induced lockdown project produced a new vision for how to build cities on asteroids. So basically, the idea is to hollow out the asteroid entirely? I mean, this just looks like they just built a station. Yeah, let's see. A wild asteroid concept would see far future humans gather up rock rubble in a massive bag made of nanofiber mesh, allowing future astronauts to build a habitat inside the loose asteroid bits as the rocks spin in space. Oh, or is it saying, like, the actual buildings? No, it says inside an asteroid. This project's art is just a way for physicists and engineers to blow off steam, set aside worldly stresses for a while, and imagine something crazy. Okay, there you have it. PhD candidate and site lead author Peter Mikulczyk, who is based at the University of Rochester, said in a statement, researchers suggest that future Manhattan-sized cities of 22 square miles, 57 square kilometers, could be built on these space rocks, just like in science fiction, assuming the base asteroid is at least 1,000 feet, 300 meters across. Okay, now is this on the... Okay, no. Again, it's like, what's an ad? What's actually relevant to what we're reading? We're taking a science fiction idea that has been very popular recently. Let's see, while the site is literally out there, the researchers emphasized all the technology is currently in place, albeit at an early stage, and that the science stands up. I mean, I guess, but again, I don't know. This just seems like conceptual whatever. Obviously, no one will be building asteroid cities anytime soon, but the technologies required to accomplish this kind of engineering don't break any laws of physics, Frank said. Yeah, but there can be a lot of infrastructure stuff. You know, again, it's like when someone says, here's how we fix the traffic problem or whatever. It's like, this would work if we didn't consider any real life infrastructure, problems or the way things are built right now. But I do like space. Let's see what's going on in space. Russian space debris forces space station to dodge, cancels US spacewalk. Something is happening in Jupiter's atmosphere, 40-year study shows. Winter solstice 2022. Oh yeah, happy winter solstice. Brings the longest night of the year to the northern hemisphere. NASA's Mars InSight lander ends mission after losing power. You know what? Sure. Let's see what this is about. Damn, one more silent spacecraft from the surface of Mars. The red planet's harsh dust is claimed in our spacecraft. NASA announced Wednesday that its InSight lander, designed to understand the geological life story of Mars, has completed its mission on the red planet. The spacecraft relied on solar power, and after 4 years on Mars, its sunlight collecting panels have built up too much dust to generate enough power to run the lander. For months now, the InSight team has been expecting the lander to fall silent. Now the robot has missed 2 calls home. Scientists last heard from the robot on December 15th. NASA will keep listening, but doesn't expect to hear anything more from the lander. I feel like there should be some way that we build these two. If that's really all it is just it got too dusty, I feel like there should be a way to clear the dust off of it. We've actually been able to do a whole lot more than what we claimed and promised to do. Bruce Bennert, planetary scientist at NASA's Jet Propulsion Laboratory in California and principal investigator of the InSight mission, told Space.com earlier this year, "I feel like looking back, this has been an enormously successful mission." InSight launched in May 2018, landed 6 months later. For 4 years, $814 million robot quietly listened to the red planet's rumblings. Unlike its rover siblings, Curiosity and Perseverance, which focus on evaluating the red planet's habitability over time, InSight was designed to peer deep inside the planet, measuring the layers from the surface down to the molten core. The mission was also meant to track current geological activity by feeling for Marsquakes. Man, with geology stuff is literally my worst field of science. Like I have always been bad at geology. Anytime I had a geology class and they're like, tell me the difference between these two rocks, I'm just like, fuck, they look like the same rocks to me. It's also why I'm very bad at histology. I just don't think I can pick up the very minuscule details. I guess I would have to study for a long time. But in fact, I took a geology class, even knowing I was bad at geology, late in my undergrad years. And I was just like, why did I do this? I mean, I did make an A in the end because it was like a 1-on-1 class, but I was like, why did I do this to myself? I know I am bad at geology. Alright, anyways. Let's see. Okay, and InSight found success on both fronts, even when things didn't go precisely according to plan. Mars itself has been surprising. It's been more difficult in some ways, and it's been more forthcoming in some ways, Bennert said. The places that were difficult, we were still able to eke out the information that we were looking for by getting more clever about the analysis and so forth. And the places where Mars was generous to us, we had things fall in our lap that we weren't expecting. And while dig, Mars was particularly challenging when it came to the InSight instrument nicknamed the Mole, officially known as the Heat Flow and Physical Properties Package. The device was meant to hammer itself down 16 feet, 5 meters, damn, pretty deep, and measure how much heat is rising up from the deep core of Mars. But no matter what scientists tried, the Mole couldn't get a grip on the soil at InSight's landing site, leaving it stuck near the surface. That is a good question. I wonder how, essentially dry and impact Mars' surface is. Like if it's all just rock or if there is some I guess for dirt and stuff you would need erosion. Like you have a little bit of dust and it's just a bunch of rock. Difficulty suggests that the ground below InSight is different than at locations NASA's rovers have previously explored, according to Sue Smrekar, a planetary scientist at JPL and deputy principal investigator for InSight. Based on our understanding of what we saw elsewhere, yes, it should have worked, Smrekar told Space.com earlier this year. So wait, did you guys choose a random site? Huh. She said she believes that the mole would have worked in one of those locations and that an adapted version could work even where InSight landed. Even without digging properly, the mole still gathered limited data, but nothing like what scientists had hoped for. Damn, isn't that the case for most experiments? It's not the heat flow that we were really after, the big prize, and that's been, for me personally, it's not the heat flow that we were really after, the big prize, and that's been, for me personally, super frustrating. Okay, wait, is the big prize the heat flow? But it's not the heat flow they were really after. Anyways, the InSight team abandoned efforts to get the mole digging properly in January 2021 after troubleshooting the issue for nearly 2 years. We knew from the beginning that this was a little bit of a tricky experiment, Baynard said. Chicken up science. But where the mole's patch of the red planet, stymied InSight? Another region of Mars was surprisingly generous. Cerberus Fossae. The region, which is scarred by faults and lies about 1,000 miles, 1,600 kilometers away from InSight, has produced far more of the Mars quakes the lander has detected than any other region. As far as we understand, there's this one superactive area and it defies the prediction of how cold Mars is, how inactive Mars is, Smrekar said. It allows us to see Mars as not a uniform and old and dead planet. Which would be good because, yeah, basically if there's no lava flow or anything, no like plates or quakes at all, then it would be just completely dead. InSight also gave scientists a better view inside Mars than any previous mission has managed to great effect. A lot of things are different than we imagined, Smrekar said. Based on the data that we had available, we had to make lots of assumptions about the interior. Now we have this hard data which gives us a much clearer picture of what's going on inside the planet. Listen to a meteor slam into Mars, see that? Nah. Oh god. Log article. InSight's data has told scientists that the Martian crust, at least to the robots near equatorial landing site, consists of 2 different layers. The top layer, about 6 miles, 10 kilometers thick, has been battered by impacts atop a deep layer about 25 miles, 40 kilometers thick. Really didn't have a clear picture about these multiple layers in the crust, Smrekar said. Even now, she and her colleagues aren't sure whether the 2-layer structure occurs globally or only in particular regions. In addition, InSight found that the core of Mars is much larger than scientists had expected. Well, that's interesting, that's good. Finding also means that the core must contain greater amounts of lighter elements than scientists thought, in particular more sulfur, perhaps as much as 15% to 20%, Baynard said. It's kind of broken our models of the core, Baynard said. When you do an experiment and get data that breaks the models, that's a real advance. It's an advance, but at the same time, it's just like, damn, our models. No one's sad to see the models go. Planets are much more interesting than our models, Smrekar said. After all, finding the strengths and weaknesses of current models is the point of any space mission. Okay, that's the end. I saw the comments still were like all the way down. The InSight team has spent recent months eking as much data out of the lander as possible as the robot's power production fell. Mission personnel arranged for the seismometer to run in 8-hour chunks, buffered by time for the lander to recharge its battery. Every different kind of Mars quake, every additional quake, it just adds another piece to the story of what's going on inside Mars, Smrekar said. Knowing that the lander caught its largest Mars quake in early May, just 2 weeks before NASA announced that the mission was nearing its end, it would be just fantastic if we could keep on. But no mission lasts forever, especially not a solar-powered Mars mission. The red planet's dust is brutal for these spacecraft, piling up on solar panels and dramatically reducing the array's power production. You know, if anything, at least make, I don't know, some sort of like cleaning rover, you know, to find these other rovers. Since it reducing the ray's power production, the dust is a double whammy, since it also seasonally fills the skies, reducing the amount of sunlight that reaches the Martian surface. Combination similarly did NASA's Opportunity rover in 2018, and now dust has ended InSight's mission as well. It's been an amazing spacecraft. It's done everything we've asked it to do and more, Bannert said. It's earned its retirement. I like to think of it as retiring and not dying. It's going to sit on Mars and enjoy the Martian sunsets for a while after it stops talking to us. Yeah, we don't need to go to comments in any article. I've gone to comments in so many, and a lot of times I just see something of just like people getting mad. You know, it'll be something like something millennials, or like something woke people, and I'm just like, this article wasn't about any of what you just talked about. What are you on about? Well, I appreciate InSight sending a selfie of itself, I guess. Oh yeah, this thing is... Okay, I was wondering what this is. Yeah, this is covered in dust. Huh. Well, let's see. Okay, probably a little shorter than I thought, but I think that was good. I think it was a good test. You know? Maybe conceptually this could be a, like, early stream sort of thing. Or just, like, start with some random article, talk about it, like, learn whatever from it, and then just be like, alright, let's play a game. I don't know, we'll see. We'll probably do it again next week, honestly. Maybe next time doing a little bit less articles and maybe playing a game afterwards and try that. But yeah, I think this is interesting. I don't want to really, you know, just keep reading more and more. Wait, baby stars burp in Orion Nebula after frantic feeding sessions. Okay, quick look at this one. I don't know, we'll leave it at baby stars burp. But alright. Thank you to everybody that came by today. Sorry the stream was so short. Maybe next time I'll try to find a better place that we can look for more sciencey level articles, but you know, it's a new idea. We'll work through the kinks of it. But alright. Hope everyone has a good rest of their day or night. And I will see you later.